Okay, welcome back to our second day, and this will be a very interesting and exciting day. I thought yesterday was terrific, and I'm sure today will be another terrific follow-on to yesterday. So today my job is to make an introduction, and so I want to introduce you to Gary Reichmuth. He represents one of our partner sponsors, so Texas Instruments is one of the companies that is generous enough to help support us at this conference. And Gary is the general manager of the High Reliability Products Group at Texas Instruments. And he will introduce Ken Arnold, and Ken Arnold is sitting here to my left, former president of TAMIS, but I will let Gary make the formal introduction. So Gary, all yours.
Thank you. It's a pleasure for us to be able to be a sponsor here at TAMIS and what it does for the industry. Just a little background on Texas Instruments. We've been supplying rad-hard parts into space back from the late 50s on the first Explorer satellite launch. We had parts on that, and since then, working with Professor Van Allen and his research, we created a custom IC, and we continue to make rad-hard parts today. So it's a very important focus for us, and it's something that we really have a pleasure in being able to sponsor this conference and what it does for the industry. So next I'd like to introduce Mr. Ken Arnold. He's going to talk about the 2019, he'll be in charge of the 2019 O'Donnell Awards Committee Chair. He's a Senior Technical Advisor of K. Arnold Consulting, and then he'll MC the next session featuring the recipients of the 2018 O'Donnell Awards. Thank you.
Thank you, Gary. All right, thank you, Gary. I want to thank you all for showing up here this morning. I hope you enjoyed breakfast. I don't know, we need to have grits at these breakfast meals rather than, but I'll take that up with the staff later. To me, this is the most important, and with apologies to Bonnie, because this is the only part of the meeting that she didn't have control over, but it's the most important part of the meeting to me. We're recognizing outstanding achievements by young investigators in medicine, engineering, science, and technology innovation, and that's really a core mission of TAMIS, is to recognize these people on an annual basis, and it's really a pleasure for me to be here and recognize them and listen to their presentations. What we do is we present them awards in each of the 4 categories that we have: engineering, medicine, science, and technology innovation, to these rising star researchers. Besides a $25,000 honorarium, each of the recipients received last night a medal with a distinct pattern reflective of the discipline, and I hope you all were here last night to see the videos. I think they did a great job with the videos. Today we're honoring 3 and not 4. We have an empty seat down there at the end. The Technology Innovation Award is not given out every year. It is reserved for non-academics, and it depends on the nominations that we receive in any year, whether we give it out or not. So I'd like to encourage everybody here who knows a non-academic who really would qualify for the Technology Innovation Award. The forms go out in February. Please, we need some good nominations for next year. I'd like to give out the award next year if we can. These 3 people have survived a very competitive nomination process. The awards committee is composed of 12 TAMIS members serving on 4 subcommittees, 1 committee for each of the awards. For the 2018 awards, there are a total of 35 nominations, 15 of which were renominations from last year. Subcommittees for science, engineering, and medicine actually select finalists to advance to an external review stage where National Academy members from outside of Texas review the finalist materials and submit their comments. In this way, we help promote our Texas researchers in places outside of Texas as well. The Technology Innovation Subcommittee conducts site visits for their finalists as a way of narrowing it down if they have more than one finalist. The awards committee reconvenes in early August to discuss external reviewers' input and recommends the recipients to the Nobel Laureates Committee. The Nobel Laureates Committee, chaired by Dr. Joseph Goldstein, reviews the recommended recipients and submits their endorsement to the TAMIS Board of Directors for final approval. So it's a rather rigorous system. It takes a while to do, and it takes a lot of people to make it work. The awards are named in honor of Edith and Peter O'Donnell, who have been steadfast supporters of TAMIS since its inception and are among the state's staunchest advocates for excellence in higher education and research. The awards are supported through the O'Donnell Endowment, and we'd like to acknowledge and thank Edith and Peter O'Donnell for establishing the endowment and the other O'Donnell Award Endowment donors for their generous contributions that make these awards possible. We'd also like to thank all the Nobel Laureate Committee and the award committee members who worked so hard looking at the nominations and selecting our 3 finalists for today. In order of presentation today, we'll have in medicine, Dr. Jordan Scott Orange of Baylor College of Medicine. In engineering, Dr. Delia Milliron, the University of Texas at Austin. And in science, Dr. Elaine Lee, also from the University of Texas at Austin. Dr. Orange will come first. So Dr. Orange is a professor of pediatrics and chief of the section of Immunology, Allergy, and Rheumatology at the Baylor College of Medicine. He is also the director of the Center for Human Immunology at Texas Children's Hospital. Dr. Orange is a trailblazing innovator in modern medicine. He has defined a new class of diseases, natural killer cell deficiencies. His work has uncovered immune deficiencies and identified secrets of these disorders to improve diagnosis and treatment for patients, particularly children. After his presentation, he'll take some questions from the audience. Dr. Orange. Okay.
Thank you again, and thank you to the Academy. It's a true honor to be here, and after hearing about the process, it's just mind-boggling to have been considered. Thank you for considering the work that we do. I look forward to trying to tell you a little bit about that. This is Texas Children's Hospital just up the road in Houston, and it's where this work has happened. Importantly, I would like to acknowledge and thank the team that has made this work possible. This is the Center for Human Immunobiology team in our lab up top. You met Dr. Mark Klein, the chair of pediatrics, last night, and grateful for everything that he has assembled to make this possible. And then, of course, I'd like to thank my family, the 4 oranges other than me, my wife Katie, and my 3 daughters. This image really speaks to me. This is a young girl outside of her home in the Pasig River, the Philippines. Probably different than the homes that we all left in coming to this conference. But what does this image tell me? It tells me a little bit about human nature, because what is this girl doing outside of her home? She's playing. She's playing despite the fact that there's not the most hospitable environment. And in fact, she looks somewhat healthy, at least on the outside. You won't see a Purell dispenser on the wall or probably a bar of soap to be found. But again, she's playing. And how is that? Because we are really set up as humans to be able to survive in hostile environments, and it was a great theme reverberated yesterday as well. So the question that has really driven me from a very early age is, why are most people healthy most of the time? And I was drawn to this question long before I knew anything about the scientific tools to get into it. What spoke to me was our inherent defenses, and that we have this immune system that is so intricate, so elegant, that keeps most people healthy most of the time. Most of us are not in hospitals. Most of us are walking down the street, going about life, and it's because we have an immune system that's fighting battles, keeping us healthy, as this little boy illustrates, doing what little boys do. He's playing in a very hostile environment, but going to be okay thereafter because he has an immune system that functions. The field that I personally fell upon is when that immune system inherently breaks down. And this is a field that we call primary immune deficiency. It's when there's a genetic inability of the immune system to provide an advantage over the environment. And arguably one of the most well-known patients ever anywhere is this young man, David, the boy in the bubble, here meeting for the first time my predecessor, Dr. William T. Shearer, who was his physician. David had a completely incapable of functioning immune system, and he survived here in Houston at Texas Children's Hospital within this highly sterile environment created by NASA. This is a family of diseases in which the immune system is debilitated. As of right now, there are over 350 named diseases in which the immune system is impaired. There's now newborn screening for David's disease, and it is uniform. Every child born in the United States is screened for this. We've also developed tools to go in and fix the genes that are broken in these diseases, really leading to banner successes in what are referred to as gene therapy, gene surgery. Also, some of the defects in these diseases have led to the design of new drugs, and there's a major drug that treats autoimmunity called tofacitinib that exists in part because of insights learned from these diseases. And they, of course, give insightful biology, and while they're rare, these children are suffering and need answers. Our laboratory really focuses in part on these diseases. It also focuses on one cell within the immune system, natural killer cells, and I'll touch briefly upon both of these topics. In terms of exploring the primary immune deficiency diseases here in Houston and in Texas, I've benefited from an amazing collaboration with a TAMIST member, Dr. Jim Lupsky, who's seated in the back, and this is something that we've dubbed locally, regionally, nationally, and internationally as the Houston Project in primary immune deficiency. And it's where we took patients that had been given up on, essentially, that had no diagnosis, and came at them from a genomic standpoint. And this has been done in two stages. The first stage of 485 individuals, and the second stage of over 1,000. The first stage has already been published, and I'll just summarize those results for you. If I may, just acknowledge the fact that this spans 22 countries, and there were over nearly 100 collaborators to this initiative, and it grows. Here are the results from the first pass, the first part of this project. There was 40% of the patients who entered into this without an answer exited with an answer, and that was inspiring. If you look at the breakdown of these, 23% of these children who had escaped a diagnosis ended up with a diagnosis in one of these known diseases. In the red sections of the pie, 8% of them ended up with a completely new condition that had not been defined before. 4% of them in green ended up with a condition that was thought to cause one thing, and because of this exercise was now appreciated to cause something completely different. And this 5% that's particularly exciting means that these children actually have what we call a blended phenotype. They have two different things that come together to make one, and when you look back, it makes sense that is these two things blended together, but one wouldn't have appreciated that a priori. And I believe that this exercise has really changed in part the way we think about these conditions. But what's most meaningful to me is this, and I won't go through it in any detail, just to say that across the different types of these immune diseases, what's shown in blue is where there's a diagnosis confirmed. What's shown in green is that there's a change in the diagnosis, but it's the orange and the red that really speak out to me, because that is where there's been a diagnosis confirmed, but a change in the way the patient was managed because of this exercise. So this genomic exploration led to a different outcome for that patient, and that's enough to get you out of bed in the morning. I want to talk a little bit about my favorite cell of the immune system, which is the natural killer cell. And this is, in fact, my license plate here in Texas. And I do like to say that if you see this car on the road, keep back 200 feet, because I'm inevitably thinking about something else. But what a natural killer cell is it's one of the innate components of the immune system that is ready to go straight out of the gate. I like to say it's consistently armed and dangerous. They are, by definition, innate lymphocytes that use germline encoded receptor systems to recognize danger, participate in immune function, and kill diseased cells. And because a picture is worth 1,000 words, let me show you a natural killer cell that came fresh from the blood of a normal person, and that's the little gray cell below, sitting on top of this green cell. And this green cell is a human cell that is infected with the virus cytomegalovirus. And that green cell shows, the green dye in that cell shows that it's alive. So actually, if we can run this video, you'll see the natural killer cell dancing over the green cell. It sits down on it, the green dye leaves, and now a vital dye that shows the cell is dead can enter, which is sitting in the liquid media. So over the process of 6 hours this natural killer cell destroyed a virus-infected cell. And these battles are going on in our body all of the time. Natural killer cells are floating around looking for problems that crop up and eliminating them before they become a problem for our well-being. So what happens if you have no natural killer cells? Well, this is combining this primary immune deficiency concept with what role the natural killer cells serve. And we've been talking about this for a while. This is sort of one recent long-winded treatise on the topic. But if you have no natural killer cells, you get sick. And you get sick with a variety of things, and particularly certain types of infections like the green cell that I showed you earlier. There are two types of what we call natural killer cell deficiency. There's what I'll term the classical, where these cells don't develop. As a result, they're low in number, or they can be missing key subsets of the cells. I like to refer to them as half-baked, and there are now 7 which are defined genetically. There's also what we call the functional category, where these cells develop, but they are abnormal in their function, and thus they're broken NK cells, and there's one that's defined genetically. But again, because a picture is worthwhile here, let me show you a picture of a patient that we are in the midst of solving right now that has one of these natural killer cell deficiencies. And he has this god-awful rash which spreads across his abdomen. And this is a rash caused by a virus that many of you are familiar with. It causes cold sores, typically on people's lips. This is herpes simplex virus, and it is very abnormal to spread across the body this way. And this is what he has, and he has this repeatedly and recurrently. And I'll show you another image which is fairly striking. Another virus that these patients are susceptible to is the common virus that causes warts. And here's a patient that has warts on top of warts. And this is really tragic and debilitating, but this is one of the things that can happen when people are crippled in this particular immune defense. So we've been certainly trying to find these patients and connect them with answers. And here at Texas Children's Hospital in Baylor, we set up something now funded by the National Institutes of Health called the NK Cell Evaluation and Research Clinic, or NEER. And this has accepted referrals from the region, from the country, and from around the globe, and is really trying to build on top of what we already know to find new answers and new insights. We've enrolled well over 100 patients at this point. The majority, of course, don't have answers, but some do, and that has been extraordinary. And then there are other journeys which are in progress. I just want to close of this topic with this one vignette. This is a family, and as you can appreciate from some time ago, what happened a few years after this picture was taken is the little boy in the red sweater got very sick with Epstein-Barr virus, one of the viruses that NK cells are useful in defending against. He was in the hospital for the better part of a year and died. Soon thereafter, his little sister sitting in front of him had the same thing happen, and she almost died and then died a few years later. The same thing happened a year later to the little boy sitting in the back. He survived. The little boy in the front had no problems. He was fine. He got the infection, never even noticed it. Had the privilege 15 years ago to meet the boy in the back and start studying him. He still had this defect, and we were able to, although we didn't have the tools at the time, but again, in collaboration with Dr. Lupsky in the back, we were able to solve this family's journey, understand it, and understand the underlying biology. And what it has become is now one of the known genes that causes natural killer cell deficiency. And as I list here, number 4, or the transcription factor, IRF8, on both alleles of the DNA is what that family has. And we've learned that a natural killer cell needs both copies of that gene in order to go through normal development, and his natural killer cells never got fully cooked. Presumably that was the case for his older sister and brother. Great insights we've learned, but more importantly, to be able to solve the journey for this family and to be able to get in front of them to try and do more for their future generations and understand that better. One of the finest days in my career was when I was able to meet with 3 generations of this family and tried to begin to explain some of what had troubled them for many years. But this rare disease, it can't stop there. It must direct our thinking, and we must follow it to novel biology. And I just want to close with a vignette that gets to how we might apply some of it. This is a disease which I've been studying for some time, Wiskott-Aldrich syndrome. And I became captivated by it by, of course, meeting a patient with it. But one of the things that puzzled me is that these children have abnormal susceptibility to herpes viruses. The kind that cause cold sore is what's causing the infection around this boy's eye, again, herpes simplex virus. And a third of these patients can get this kind of thing, and then a third can die from blood cancers. We began studying it, and we found some time ago that the natural killer cells here are broken and defective. As I showed you in that first video, the natural killer cell must interact with a disease cell, recognize it, and then use this specialized machinery it has to line up, aim towards it, and destroy it. Essentially, a whole series of bullets, and about 200 of them to be exact, are lined up and directed towards that disease cell. You can see on the top cell, there's that big, thick area of blue, and that's the structural framework. That's the cytoskeleton of the cell, what we call actin. And behind that, in green, are these bullets aimed up to destroy this cancer cell sitting below it. In the patient's cell below, you see this very fine, inadequate ring of blue, this inadequate structural framework. And the bullets in green are essentially all over the place and not really set up to destroy the tumor cell on which this is seated. Gave us some insights as to the role that this cytoskeleton may serve in leading to the progress of lining up the destructive machinery to destroy a diseased cell. That's been a theme of research in our laboratory for some time, and we've now learned that there are many more steps, linear steps, that lead to the recognition of a diseased cell to its destruction. And we've, over the years, been defining more and more steps, been subdividing them. And if you apply Zeno's paradox, we'll be able to keep going with this, hopefully indefinitely, with solid funding from the National Institutes of Health, but we'll see how that goes. But again, I want to show you what this looks like. So here's a natural killer cell in green on a tumor cell in yellow. The red bits are of these bullets, what we call lytic granules, and they've conglomerated, we call it converged, and moved towards the diseased cell. If you can run it again, there's a 90-minute video sped up for 12 seconds. They converge, they polarize, they hang out for a little bit, and they start shooting out on the diseased cell and destroy it. And you see the blue dye go in, which shows its death. So it led to the question that I'd like to leave you with, which is, why do they do this? Why are they set up from an evolutionary standpoint to take all of this machinery, pull it together, and target it towards one offending event? Well, because these are part of the seek and destroy system, I would propose, within our bodies to eliminate and eradicate danger before it becomes a problem. So there are, sadly, cancer cells in our body popping up all the time. And what natural killer cells can do within the context of this one diseased cell in an otherwise healthy tissue is go find it, recognize it, eliminate it, and then get about business. But what evolutionarily they're not set up for is this context where I would say it's too late, where that one cell has evaded recognition, and now we have tumor cells, a large tumor. They're not set up for that. And what they'll do is they'll try to do their best. They are serial killers. They will go on and recognize as many cells as they can, but again, this is really not what they're configured for. So why do they become this sniper, if you will? Well, they utilize the machinery, and I think there's a reason for doing it. They take these bullets, these lytic granules, and they move them in using a motor on the cytoskeleton to converge. They pull these centrally, And then they polarize them along the cytoskeleton using something called the microtubule organizing center towards the disease cell. So you get all of the bullets on the disease cell. And again, because they're on these seek and destroy missions, and one of the reasons that I believe they want to do this is they want to protect otherwise healthy cells around them. They want to protect innocent bystanders and kill the disease cell. And we got some insights as to how this convergence process, this conglomeration of the bullets occurs from another rare disease called leukocyte adhesion deficiency, where the sticking mechanism is broken. And what we found there is that you'll see on top the red, the lytic granules, the bullets. Normally, when it sticks to a cell, they're all converged and pulled together. And if you don't have that sticking ability, that leukocyte adhesion molecule, they're all over the place. So that's the key signal for doing that. And what we wanted to then ask is that, can this convergence process, can this positioning that allows us to do this surveillance function, can it be controlled and can it be exploited? So if you can play, so the video that I want to play first is the one called ADCC. So what we've taken is a tumor cell and we've coded it. It's usually resistant. We've colored it in green, and we've coated it with a therapeutic that recognizes tumors called rituximab. And what happens there is the natural killer cell will go recognize that one, and it will destroy it. The one that's not coated with the therapeutic is in yellow, and that is our innocent bystander, and it will be left alone, in theory. And so if we go on to the other one, though, but we block this signal that allows the bullets to come together, well, in this case, what might happen is that the bullets will release all over the place and not only kill the disease cell, but they'll kill the innocent bystander. We wanted to try and figure out how to look at this experimentally to see if it was true, and we had to develop an approach that we like to call UGATM, or ultrasound-guided acoustic trap microscopy, to be able to pull together different cells on the snap of a finger. So if you run that video, you'll see UGATM. What is it? It's an ultrasound in a small micro well that is tuned to a particular frequency so that when you turn it on, you can see the cells come together right in the middle. And that's what this has allowed us to do. So on-demand approximation of different types of cells to try and simulate what might be going on in our bodies. So I'll show you what the experiments look like, and I just have two videos to run. So the first is if we have the tumor cell in green coated with the therapeutic, and in yellow is the bystander. And what you'll see is the natural killer cell here, the small one in red, goes and sits in between those. And despite being next to a bystander, even sitting on top of it, it only kills the diseased cell. But now if we try to control the positioning of the granule, and we block the convergence, we block the bullets being able to come together, the natural killer cell will go sit right in between the two. And bang, it non-discriminately kills both. So why am I mentioning this, and what potentially can we learn from it? Well, this is what normally happens. So the natural killer cell as the sniper, doing what it does, working in this context when we have a tumor, when it missed its boat, when it's too late. So if you can play this video, the natural killer cell goes into the tumor environment, tries its hardest, converges its lytic granules, its weaponry, goes and kills one cell, and then goes and kills another. But what I get really excited about, what we've learned from these diseases, is how we can potentially exploit some of this biology to gain therapeutic advantage. To gain therapeutic advantage when the natural killer cell is done doing what it should have done, missed the opportunity, but we want it to do something else. So perhaps to turn the sniper into a grenade. So if we can control granule positioning, hopefully what we can do therapeutically is get the natural killer cell to go into a tumor environment, degranulate, shoot its weaponry non-discriminately, become a grenade, and that one single cell can now overcome many challenges and hopefully not only just get rid of other cells in the tumor, but cells that are in the process of escaping the immune system, other cells that support a tumor that might provide a growth environment for the tumor, and we're really optimistic about this concept and trying to advance this within the growing field that many of you may have heard about in the news of cellular therapeutics, where these cells are now being utilized to try and treat and cure cancers in patients. So in conclusion, natural killer cell deficiency is an emerging primary immune deficiency with a number of genotypes. The convergence process in natural killer cells promotes this directed killing and the potential to exploit the positioning therapeutically to overcome what I see as the evolutionary purpose of NK cell cytotoxicity can perhaps help us unleash and utilize this process of bystander killing and turn nature's snipers into grenades. So with that, I'd really like to thank Tamist for recognizing this work, and thank you, Texas, for being such a wonderful environment in which to make it all possible.
Thank you, Dr. Jordan. We have time for one or two questions, if there are any.
This is an engineer's question, if you will. In my body, typically, how prevalent are these NK cells in any given element of my body?
Yeah. So they are actually 1 in 10 of the lymphocytes that are floating around in your blood right now, on average. They also collect themselves within certain organs of your body. They are really present, armed and dangerous, and ready to step into defense. So they are a key part of who we are and what we do.
Okay, Adam.
Just 2 quick questions. First of all, thank you. Great work. First is, you would imply, or it seemed like you implied that this was some of the PIDD and NKC deficiencies were inheritable. And the questions are, is there a genetic testing program in place and counseling or educational processes that would go with that? And then the second, or follow-up question is have you also identified idiopathic forms of these same diseases?
So the first question is the easier one, in a sense, because these disease genes are being increasingly recognized. They're being proven as causative, which is harder because the biology has to be done. But then once they find themselves into the OMIM database, Online Mendelian Inheritance in Man, as well as diagnostic laboratories that do sequencing are able to provide these results to patients, to genetic counselors who can integrate some knowledge and the literature and be able to counsel the individuals and hopefully lead to treatments. And so this is happening, and some of the conditions that we've contributed to even just a few years ago are now unfolding in clinics with patients. As far as idiopathic forms, idiopathic is one of these terms that really rubs investigative physicians the wrong way, and it's really kind of course, what makes us idiots and when we have to use that label because we can't figure out the answer. But the reality is that we do have forms we don't understand, and hopefully through investigation, we'll be able to connect them with specific answers. And we do follow a number of these patients, but we're working hard to try and understand what causes it.
Okay, thank you very much.
Thank you.
Thank you. Good presentation.